Hello, this is our Bio375 Lesson 9 video. So I'm just going to go right into it. I'm Joshua Allred. I'm doing the first question. It says to diagram DNA replication here, and I'm supposed to include all these terms. So let's hope you can see that there. And as you can see, these parallel lines are in this bootstraphic relationship. And we have DNA primase, which is creating this RNA primer. We have the origin of replication, DNA helicase, which splits the DNA. The primers, as I pointed out, and we have two leading strands, DNA polymerase 1, which has this little double guy here, and he kind of replaced this RNA primer. And then DNA polymerase 3 alpha, which is synthesizing the DNA from 5' to 3', and several Okazaki fragments. All right, now I'm going to pass over to Jacob.
There we go. So I'm going to be going over number 2, which is just fill in the blank. So we'll go down the list. Put my computer over here so you're facing more of the camera. So we have DNA primase, the protein complex that synthesizes primers in eukaryotes. Helicase, the protein promotes separation of the AT-rich sequences within ORIC. The epsilon subunit, the subunit of DNA polymerase 3, contains proofreading activity, and really you're going to be seeing this throughout the whole thing, is really polymerase. The whole set of polymerases are kind of like the mana that really keeps everything going in like reproduction. DNA polymerase 3 alpha is the protein that synthesizes the leading strand in eukaryotes. TUS, or TUS protein. This protein displaces DNA polymerase from the chromosome to terminate replication. DNA polymerase 1, this one has 5' to 3' polymerase activity and 5' to 3' exonuclease activity as well. Topoisomerase type 2 is the protein that introduces negative supercoils in the DNA ahead of the replication fork, which helps it unbind, so you can actually see that breaking apart that my partner beforehand was showing. The single-stranded binding proteins, SSBPs for short, prevent DNA strands from annealing during replication, which is just them gluing themselves back together, which would be really annoying. It's kind of like working with sticky tape and then you try and get it off one finger only to have it on the other finger. And that is like the anti of that. And then DNA ligase. The enzyme seals Okazaki fragments in a continuous strand of DNA. Then 3, the question concerns the enzymes involved in DNA replication. The first question is list 5 enzymes, prokaryotic or eukaryotic, so we can pick from both groups, that consume adenosine triphosphate during DNA replication. And the list that we came up with was DNA primase, helicase, topoisomerase type 2, ligase in general, and DNA proteins. I spelled protein correctly. List 5 enzymes, prokaryotic or eukaryotic, that consume D-ATPs during DNA replication. DNA polymerase 1, DNA polymerase 3, DNA epsilon, DNA poly alpha, and DNA polymerase beta. And all of those would also go with the next one, which would be 3' to 5' exonuclease activity, which would be all of those. Indicate one enzyme, prokaryotic or eukaryotic, involved in replication that uses DNA as a template to synthesize RNA. DNA is polymerase 3. Well, what it does is it takes those little like primer, those heads off of the RNA and then puts in the DNA, and then indicate one enzyme that uses RNA as a template to synthesize DNA is telomerase. Yeah. So now I'm just going to hand this on over.
Hello. So for number 4, we are basically running a PCR reaction, slightly different. So for the first one, you are given the DATP, DCTP, DGTP, and DTTP. And so for that one, the whole strand is going to replicate. So the kith strand for that one is TATCG, TCCT, GGTA, GTCAGGAAA, TTCGT. And that's the longest one. So that's, yeah. And then the kith strand for the second one is just TAT because you have the CMP there, which makes that stop. And so for C, because you have the UTP, and we're synthesizing DNA, and the first letter is a T, nothing is going to form. And then for the last one, you have the GTP, that isn't deoxyribose. And so that one is just going to go to the first G, which makes the kith strand TATC. And then for number 5, proteins that are found in prokaryotes are DNA gyrase, DNA polymerase 3, the alpha subunit, DNA polymerase 1, pCNA, DNA primase, the TUS, and DNA A. And then for proteins that are found in the mouse musculus, you have telomerase, DNA polymerase alpha, the MCM helicase, the CDC6 compound, and DNA polymerase epsilon. For C, none of the proteins there are involved in chromosome compaction. And then for the proteins involved in the initiation stage, you have the CDC6, the MCM helicase, and the DNA A box. Then for E, which of the proteins are involved in the termination stage? And that is simply TUS and telomerase. That is our video.
Choose.